Taste and smell. Okay, interoreceptors detect chemical changes in the body. That's within the body. That would be a buildup of carbon dioxide. It would be high glucose. It would be interochanges with chemo. So it would be like high glucose, high carbon dioxide, high sulfur, whatever. Exteroreceptors are receptors that are getting their stimulus from outside the body again. Extra, outside. Taste responds to chemicals dissolved in food and drink. Smell responds to chemical molecules in the air. Now the thing about smell, as I said earlier, they have to dissolve in the mucous membrane in the olfactory system or the nose or the smelling system before they can be, before that sense can work. Taste, called gustation. So you have the taste buds. This is your taste bud. And it has cilia on it. Most of your epithelial cells have cilia on them. So that's your taste hair. And then you have the taste cell, and you probably have, 50 or 100 taste cells in each taste bud. And then the supporting cell, and then the pore is where the chemical's going to get in. Surface of the tongue, taste bud is the whole thing. Taste pore, hair cells which are stimulated, the taste cell, supporting, and urethra that sends it off. And so that you can actually have the taste. That's where the action potential is.
I have a question. Like, doesn't go to that, but so for people who get their tongues pierced, like that thing, doesn't it ruin their taste buds then?
No.
No? Well, I know people who have the tongue pierced and they say no, but if you're saying it's all taste buds, wouldn't it affect?
Well, you have
many of
tens
them?
of thousands of taste buds.
I had mine done 3 different times. I still think everything's fine.
Yeah, that's what my friend has said too, but I just always feel like I don't know. Okay.
I can't even begin to address that.
But is it a scientific response? Because did you test how much you tasted?
Test how much you tasted before you
exactly.
Actually,
Because
I think that I had less taste buds before then. I used to think everything was very, very bland, and I only actually enjoyed bland food, but now I enjoy
Open taste buds.
I don't know.
So that's a recommendation to everybody.
Get the tongue pierced.
Right.
Okay. Going on.
That's going to do that more.
Yeah, right. That's right after I lose the bed who's to get the first hearing. And I'm nowhere near on that one. Okay, the lumps on the tongue are called the papillae. You have the fungiform on the anterior surface, and those go via the facial nerve. And I believe that's cranial nerve 7. There's the circumvallate, and the folate are on the sides, and they go through the glossopharyngeal nerve. Now, depending on how, that's another one of the cranial nerves. I think that's 10. I'm not sure. No, 10's big. 9. Okay. I don't remember it off the top of my head. But it doesn't mean I won't ask it on the test. So you have 2 different nerves sending taste into the brain. Facial, glossopharyngeal nerve. Medulla goes to the medulla, goes to the thalamus, and goes to the cortex of insula. The somatosensory cortex of the parietal lobe and the prefrontal cortex. The insula, again, is part of the limbic system. And how you feel good when you got something that tastes good? That's part of it. That's part of it. Taste cells of taste buds. Specialized epithelial. They behave like neurons. We'll sometimes call them a neuroepithelial cell, which is not quite right, but behave like neurons by depolarizing, producing an action potential. These are your taste cells down here. It's starting to ring again. These are your taste cells down here. The whole thing is the taste bud. Release neurotransmitters. Microvilli come into the contact with the chemicals, the hairs at the top of the taste cell. Each taste bud has taste cells sensitive to each category of taste. What are the categories of taste? Sweet, sour, salty, bitter, umami. Ever heard of umami?
Ooh, mom.
Not It's named umami because it was discovered by the Japanese, and it's savory and meaty type taste. And it's a glutamate receptor, and it's really, in the spices and that, the monosodium glutamate. That's what gets this receptor to fire. Like the capsaicin would get the heat to fire. This is a monosodium glutamate, which is not good for you, but what the heck, it tastes good. Okay, so 5 categories of taste. Salty. Sodium. Through the ion channels, it opens calcium channels, and you have the synaptic membrane, the neurotransmitters released, and off it goes. Sour. Hydrogen. Salty is sodium, sour is hydrogen. That's what causes it to release. Again, it opens calcium channels, causes a neurotransmitter to be released. Sweet and umami. You have sugar binds. And then there's a G protein. And what does a G protein do? Alpha, beta, gamma. The G protein breaks up and starts a second messenger. The second messenger goes down, and it doesn't open calcium channels, it closes potassium channels. Same deal. It causes depolarization, and off, then you hit the synaptic cleft, and you have your stimulus. Then bitter, quinine. Again, a G protein, a secondary messenger. Calcium this time is not from the extracellular, but it is from the endoplasmic reticulum, which stores a lot of calcium in this, and especially when we get into the muscle cells. Then you have your neurotransmitter. The bitter is by far the most sensitive. They hypothesize or speculate it's probably because toxic substances are generally bitter and might keep you from ingesting some of them. Salty. Sodium enters the taste cell and depolarizes it. Sour is hydrogen. Bitter, sweet, umami, sugar or glutamate. Bitter, quinine. And then you have your G protein and your calcium. Sweet and umami is G protein and potassium. Sour is hydrogen, salty is sodium. That causes depolarization or the action potential. G proteins and second messengers. G proteins are called gustidins, In the sweet system, it produces adenylate cyclase, CAMP. Other molecules, it may activate IP3 and DAG, and we will touch on that as we get in. Smell, called olfaction, located in the nose, nasal cavity. Okay, you have sustentacular cells, hydrolyzed, hydrophobic, volatile odors. This doesn't help your sense of smell. It keeps you from inhaling toxic stuff. It protects. And then you have the basal stem cells in the olfactory system, and they're constantly replacing the receptors that are damaged. You figure I think it's every 2 months, 6 weeks to 2 months, you replace all your receptors. So here, your nasal cavity, you have your cilia, you have your receptors, you have your neurons. They go up through the cribriform plate to the glomeruli, and then off to tufted cells into the secondary neuron, and then up into the brain. The olfactory receptors are bipolar, which means they have more than one set of dendrites projecting into the nasal cavity. Proteins in the cilia bind to the odors, and the proteins in the cilia are in the mucous membrane. The odors get into the mucus, and that's where it's picked up. 350 genes code for 350 different receptors. Well, you can smell a lot more than that. The brain will put a pattern to it. Olfactory receptor. They're G-protein coupled, which means what? You need a secondary messenger, G-protein. G-protein is coupled, it releases an alpha, beta, or gamma particle, which goes and opens up and sends a secondary messenger to open the sodium and potassium channels. Odorant, G-proteins, adenylate cyclase, ATP goes to cyclic AMP. Cyclic AMP causes this potassium or calcium channels to open. Typical secondary messenger type system. Olfactory neurons are unmyelinated, which means are they faster or slower? Slower than the myelinated ones. Synapse on a glomerulus in the olfactory bulb right up inside the cribriform plate. Each olfactory receptor synapses on a particular glomerulus. A flower is a complex number of odors. And what happens is the brain interprets all these different odors that are coming in, and you say, oh, that's a rose, or that's whatever. But the only one I can think of is smell for. On the other hand, walnut smells good, especially when you're working with it. But it's coated. A flower may excite all these different glomeruli for different receptors, and then the brain interprets it as the type of odor that you get, that you sense. Neurons from the olfactory bulb synapse in the prefrontal cortex. Medial temporal lobes, hippocampus, and amygdala. Again, the hippocampus and the amygdala are very important in the emotional aspects, part of the limbic system. You've heard of aromatherapy? Mm-hmm. This is where it works. You've heard of pheromones. You familiar with pheromones? Pheromones are a chemical odor stimulant. In mammals, it would be a pheromone would be the odor like a dog in heat that a male dog can smell. To a certain extent, humans have the same thing. Though I think we use, it's part of what women or men or whomever would use it for cologne or perfume to try to mimic.
What's associated? I know there's something that translates smell to memory, and that's what smells.
That's in the hippocampus and the amygdala. Amygdala also. Memory and emotion. Okay? Vestibular, okay, so there, we're done with, pretty much with taste and smell. Any questions on that? What's happening with taste and smell is the brain, you've got all these different taste buds, they're sending patterns in, the brain is interpreting this different type of pattern that says, this is salty, this is peanut, this is salty, this is cashew, this is sour, it's lemon, this is sour apple, this is sour whatever. The brain gets all these different firings from the different neurons, and then it interprets them. And part of the interpretation is, from the hippocampus and amygdala, it's part of the memory, because you have to have an idea of what it is before you can interpret it. And if you've never tasted sweet or sugar before, you're not going to know what it is until you try it and know what it is. So the brain is, again, taking all these, your external environment is literally changed into neurons and into action potentials into the brain, and then it's the brain that actually interprets it. And it does a pretty good job. Okay, vestibular apparatus and equilibrium. Vestibular apparatus, we talked about it in lab. We looked at vestibular nystagmus. The vestibular apparatus has the otolith organs, utricle and saccule, and it has a semicircular canal. The utricle has hair cells in it. And as you move forward or backwards horizontally, these hair cells, these cilia bend. And you get the feeling of motion, either forward or backwards. The saccule is up and down. That's what's bending the hair cells. So here's your utricle. And this is not great, but it's it's a little more horizontal than the saccule, which is a little more vertical. But the utricle with the cilia and the hair cells is going to, again, look at or sense a motion horizontal. Saccule is going to sense vertical, and the semicircular canals go in all three directions. Kind of like that guy that does the balloon. The long balloons, they all go in, he makes the crown, it all goes in three directions. That's your semicircular canals. They're filled with endolymph. Endolymph starts moving, or doesn't move when you move, and then after a while it starts moving. That's where we saw that vestibular nystagmus on last Thursday. But let's go on. Inner ear. You have the bony labyrinth surrounding a membranous labyrinth. Between the two is perilymph. Perilymph is different than endolymph. Perilymph is pretty much like any extracellular fluid. Inside the membranous labyrinth is a fluid called endolymph. The endolymph is the only fluid in the body that's real high in potassium, naturally. So here's your membranous labyrinth here. The bony area around it is the bony labyrinth. So when you're looking inside the bony labyrinth, you're seeing the membranous labyrinth. Sensory hair cells. Modified epithelial, 20 to 50 hair-like extensions called stereocilia. Then there's one they call the kinocilium. So you have all these stereocilium, and then you have the kinocilium. When it pushes on the kinocilium, the nerves are stimulated, hyperpolarized, or I'm sorry, depolarized. When it pushes away from the kinocilium, they're hyperpolarized. You don't get the action potentials. Here you get the action potentials. This is how the body tells it which way it's moving. Hyperpolarization or hypopolarization. So remember, we started somebody spinning. So they were, so what happened is they were pressing. This was pressing. Until everything started spinning at the same rate. And then you're back to as if it was at rest. There's no change. Nothing's getting pushed one way or the other. Until you stop. Then you stop and the endolith keeps spinning. So it goes in the opposite direction. That's why your eyes moved. And when you were spinning in the opposite direction of the spin, and then when you stopped, in the direction of the spin, because of the way the endolith was moving. Okay, when it bends toward the kinocilium, it opens, the potassium channels open, it depolarizes. When it bends away, it hyperpolarizes. That's what we just went over. And this is interpreted in the brain as code for direction of the spin. This is in the semicircular canals. The utricle, again, provides information about linear acceleration, and the saccule about vertical or acceleration, horizontal or vertical. If you're going up and down in an elevator, and once you start moving, you don't feel it. When you're accelerating, you feel it. Or, taking off in a jet, or in a car, whatever, you feel it, and then you don't. Specialized epithelium called the macula, and houses the hair cells. The stereocilia are in a gelatinous otolithic membrane on the semicircular canals, but it just means that the endolith, they wave, like a sail. They wave as the fluid moves along. Okay, this is an otolithic. You're looking at if the lady, when she bends her head, the stereocilia are bending, the otolithic membrane sags, you tell that you're bending down, the gravitational force. Again, the hair cells in the endolymph, they're sensory. And the way they move gives you the sense of direction. And that's really kind of the take-home message. Saccule, the utricle, and the semicircular canals. They're in 3 planes, so you can tumble, you can spin, tumble, cartwheels, or circles. And there's one in each plane to detect that. The base of each duct is the ampulla. The hair cells are embedded in the ampulla. The stereal cilia are stuck to a gelatinous cupula, or let's see what we got here. Supporting cells right at the bottom. So you have the endolymph, the cupula, the hair cells are here, sensory nerves. So the hair cells are coming into the nerves. When they bend with the endolymph moving, then you have the sensation. You have the action potentials. And this is all over, all through the body. You have this type of not endolymph, but you have the cilia that are attached to nerves, that create an action potential, and gives you an idea in the kidneys, in the bronchi, in the semicircular canals, in the nose. Not the movement in the nose, but they are sensory. Vestibulocochlear nerve synapses in the medulla. Now, one thing about this is the medulla then sends it to the oculomotor area. That's why your eyes move. That's why when you're spinning, your eyes are trying to track it. And you keep your balance. And then it goes down the brain, spinal cord, again, to adjust body movements. You're spinning, you're standing up, you're turning, and your brain is trying to keep everything balanced so you just don't fall flat on your face. Obviously, somebody like Christina Noguchi has it very fine, very finely tuned. Nystagmus. Person's body is spinning, the eye movements are toward the opposite direction. When it stops, again, like we saw in the lab, the eye movements went back to the same direction as the spin. And this is called nystagmus. This is because the endolymph is still moving in the semicircular canals. And it's bending the cilia. You're still getting the impulses. And again, the nerve is just reading the impulses. The brain, is just reading the impulses. You may be stopped. The brain's saying, hey, I'm still moving because I'm getting these impulses, and that's what's going to the ocular motor nerve. That's why your eyes are still trying to move and trying to adjust. Cause equilibrium. You spin it up. You ever stand up and try to walk in a straight line? This is it. Dizziness, pallor. How many of you have been on the tilt-a-whirl or something like that at a night? Too old? You know what a tilt-a-whirl is? At a fair?
Or the spinny things that they have, basically, like the teacups at the beginning.
Yeah.
I
Mr.
had
Cody.
that Frontier Village many years ago.
Well, that was, I grew up in little country fairs in Minnesota. Everybody had a tilt-a-whirl. What's that? Minnesota? Yeah, you betcha. Dizziness, pallor, sweating, nausea, vomiting, motion sickness. That's where it is. Okay? Vertigo. Vertigo is a dizziness. It can be caused by an ear infection. Too much fluid. Meniere's disease. It starts with ringing, tinnitus, or ringing in the ear, too much fluid. This would be pretty classic of Meniere's. Okay, sound waves. Now, even now, you're still going to have this variability. You're going to still have long hairs and that. We'll get into that. Frequency is measured in hertz. Remember when we looked? Frequency. Let's see if we've got it. Frequency is how close together the sound waves are. This is a high frequency. This would be a low frequency. Amplitude is how high they are. So this is pitch. This is loudness. Amplitude versus frequency.
What do you mean? The pitch is the distance between each.
Well, the closer they are, the higher the frequency, the higher the pitch. The larger the amplitude, the louder the sound. Intensity or loudness is measured in decibels. Decibel, the lowest thing that you can hear, a whisper, would be a 0 decibel. And then essentially for every 10 times, you would raise it to the power. So 10 is 10 times, 20 is 100 times, 30 is 1,000 times. So decibels go up very quickly. So here you have the external, you have the earlobe, the external auditory meatus. This captures the sound. You come into the tympanic membrane, which is the eardrum. This vibrates. Then you have the bones. You've got the outer ear, middle ear, inner ear. And then, so you have your hammer, anvil, stirrup. You've got 3 bones in the middle ear that conveys the vibrations to the oval window. Oval window, does it mark it here? This is your oval window right here.
The auditory ossicle is the oval window, isn't it?
No.
Under the bone.
Auditory ossicles are the bones. These are the bones.
Looks like they labeled it round.
No, the round window is another part. It's at the other end of the cochlea. What happens is the bones go up to the oval window and they press on the oval window. Vibrations of hearing go in through the oval window. And then your inside of the oval window is filled with a perilymph. And fluid doesn't compress. So now these vibrations go into the inner ear, into the cochlea, and there are cilia.
Yeah, even the oval window is at the base of the bone that is in contact with the cochlea. The cochlea.
The oval window is right here. This is your oval window right
Exactly.
there. And your round window is over here. But what happens is, because the fluid doesn't compress, and if you ever had an engine that got water in it, it was running when it went into the lake. It didn't happen to me, but it was close. Water doesn't compress, so if water gets into an engine like that, the engine breaks before the water compresses. You break a rod, you break a piston, something like that, because it will not compress. When you have something where you're a tank sprayer of that, you're pumping it, the only thing that's compressing is the air, and then that pushes the water out. But the water itself does not compress. The same in the inner ear. It's filled with fluid, it does not compress. So you have the oval window imparting vibrations that go into the inner ear, and something's got to give. So it's the round window. That's what gives at the other end to keep it from the pressure from building up in the ear. So it goes in through the oval window. It essentially goes out through the round window. Sound waves funneled by the pinea or auricle into the external auditory meatus, into the channel, and through the to the tympanic membrane or to the eardrum. The cavity between the tympanic membrane and the cochlea is the middle ear. 3 bones, malleus, incus, stips, hammer, anvil, stirrup. Vibrations are amplified along the bones. Now, the stapes is attached to the oval window, like I pointed out here. And that's what transfers the vibrations into the inner ear. Now, the other, you have a scrupedius muscle. Let's see, the scrupedius muscle is here. This is a muscle, you see it's connected to the stapes. I wish I had mine. It's connected to the stapes. If you get a loud noise, the scrupedius muscle contracts, pulls the stapes back away so not all this loud noise goes into the inner ear. It's a protective thing. If this muscle is weak, if the noise is too sharp and the muscle doesn't have time to contract, this allows that vibration to get into the inner ear. It can damage the nerve receptors in there. Hence, you wear ear protection when you're shooting. Now, you don't wear ear protection when you put the iPod in your ears and turn it up. And I think we're going to have generations of poor hearing simply because of that. Because you are degrading the nerve cells in the inner ear.
So the muscle moves the bone away from the oval?
Or it impedes the vibrations through the bone into the oval window so that you don't get this loud noise into the inner ear. It's protective. It's a protective feature. The cochlea. Cochlea looks like a snail. This is your cochlea here. So the cochlea and the bony labyrinth. This is where the hearing, this is where the rubber meets the road. This is where your nerve endings are. This is where your sound is, the vibrations are made into action potentials and sent off for interpretation. 3 chambers. The upper chamber is the scala vestibuli. The lower chamber is the scala tympani, and the middle chamber is the scala medium. These chambers are filled with paralymph. Remember, paralymph as opposed to endolymph. Paralymph is much more like a regular high sodium, low potassium extracellular fluid. But again,
The endolymph, I'm sorry, is the high potassium?
endolymph is high potassium, and that's in the scala media or the cochlear duct. The rest of it is filled with paralymph. And we'll, the scala media is in, here's your scala tympani, paralymph, cochlear duct, and here is your scala media, and here's your outer one, your scalar vestibula. Or I might have that backwards. Upper chamber, middle chamber, lower chamber. Vestibuli, media, tympani. These two have paralymph. This one has endolymph. This is, you can see how they would have sliced it right down through the top. So there's the top of the cochlea. There's the bottom where the nerve, vestibulocochlear nerve, facial nerve 7 comes in. Vibrations from the oval window. The middle ear displace the paralymph and the scala vestibuli. Vibrations pass in the cochlear duct through the endolymph. Pass from the paralymph next into the paralymph or the scala tympani. Vibrations leave via the round window. So they're coming in through here, through the oval window. They're going round and round, and they're leaving out of the round window here. Now, along the cochlea, as we looked at earlier, you have different frequencies of sound. So as it winds through the cochlea, the sound, the frequencies hit it in different areas. And this is how you tell the difference in pitch, is where they hit. Lower frequencies travel further down. They're more spread out. Here. Here's your higher frequency in the blue. I'm sorry, your lower frequency in the blue is spread out over the entire duct. Your higher frequency is in the red. You see where the sound waves are hitting in the duct? All at one end.
What, again, that sounds stupid inside, but what measures the waves in the ear? What, if the nerve sensation, how strong the nerve catch the stimulus?
Well, what measures the waves in here are cilia. There's hair like cilia in there, long and short hairs. And we'll get to that in a minute. In the air out here, you would have to have some sort of electronic instrument. The sound waves can be measured for their frequency in the same way you would measure an electrical wave. But in the ear, it's still, they're measured by where the frequency and that are measured by where they hit along this duct. Okay, so you have the higher ones hitting at this end, the lower ones hitting further out. And we'll go through that a little bit. Sensory hair cells are located on the basilar membrane. Sensory hair cells are located along here, along the basilar membrane. The inner hair cells form one row, runs the length of the basilar membrane. Each is innervated by about 10 to 20 neurons. The outer hair cells are arranged in rows. There's only one row of inner hair cells. The outer cells are arranged in rows. They're innervated by motor neurons. They shorten when depolarized and lengthen when hyperpolarized. The stereocilia are in the tectorial membrane. This is your tectorial membrane here. This is your basilar membrane here. Sorry, but there's your tectorial membrane. When sound enters the scala media, the tectorial membrane vibrates, bending the stereocilia. So what happens, is it's where these stereocilia are vibrating. Outer part, inner part, and our latter part. And then they will send different signals off. Inner hair cells from sound waves enter the scala media. The tectorial membrane vibrates, bending the stereocilia. Opens potassium channels, potassium runs in. Remember in the endolid, potassium is much higher. Releases glutamate into sensory neurons. This is the organocorti in there. So here's your tectorial membrane, scalar tympani, upper, middle, lower, cochlear duct. Again, this is a different slice from a different direction of your cochlea. But here's your basilar membrane again. Right down here, your tectorial membrane's on the top. This is what starts vibrating. Here's your inner hairs. There's your outer hairs as part of the tectorial membrane. They're connecting to nerve fibers into the vestibular cochlear nerve, and then off through the facial nerve 7. Hair cells located closest to where the vibrations are, displaced in the scala media, are stimulated more often. Outer hair cells magnify this effect, which allows us to differentiate between pitches. So here's your inner hair cells. And if you go back, tectorial membrane, it's where they are being activated, where they're being, where the receptors are on the hair cells. Enter the scala media, the tectorial membrane vibrates, bending the stereocilia. hair cells closest to where the vibrations, the higher frequency vibrations are more toward the outside. The lower frequencies go all the way into the cochlea. So you have the one row of inner hair cells, but then the longer outer hair cells are much more sensitive to the vibrations. This allows you to discern pitch much better. So the neural pathways, you've got the vestibulocochlear nerve, the medulla oblongata, inferior cochleus of the midbrain, the thalamus, then into the auditory cortex. Tonotopic areas represent different sound frequencies, different tones. That's your neural pathway. So here's your cochlea, and as we said, here's your high tones. High tones have a high frequency. They don't get far into the cochlea. Your mid-tones in here, and your low tones in here, because this is how they vibrate. The wavelength is longer, it gets further into the cochlea before it's stimulating the hair cells. Then it goes in, what's that?
I'm sorry, tones and pitch is all
Same thing.
synonymous.
Same thing, yeah. So it's going to come out of the cochlear nucleus, it's going to go into the midbrain, auditory cortex, and then up into the temporal zone. We talked about conduction deafness. Okay, first of all, do you have questions on the hearing? Comes in through the outer ear. It's the tympanic membrane. Goes into the middle ear with the bones. They vibrate the oval window. The oval window goes into the perilymph and the endolymph. It goes into the cochlear apparatus. Inside the cochlear apparatus, you have the hair cells that vibrate depending on the frequency. It moves through, it vibrates the hair cells, and then exits through the round window. You have the short here, or inner hair cells that are one row. They detect the cell. It's amplified by the long hair cells, or outer hair cells. Innervated by motor neurons, they depolarize. They shorten and depolarize, hyperpolarize, they elongate. That's how you tell the different pitches. Goes into the inferior colliculus, into the thalamus, and into the auditory cortex of the temporal lobe. Conduction deafness. They don't get, if it doesn't get into the oval window for some reason, either the middle ear is full of fluid, the outer ear is blocked, injured, that's a conduction deafness. That's where a hearing aid can help. It can force it through. And it's across all frequencies. Just the sound isn't getting, the vibrations aren't getting in there. Now, if you have the sensorineuroperceptive, that's nerves. They're not conducting from the cochlea to the auditory cortex. Those are not all or nothing. There will be pitches you can hear, pitches you can't hear. Frequency or pitch. This is what is damaged due to the loud noises in your iPod, things like that. I'll let take my boy's iPod and step on it. Maybe helped by cochlear implants. They essentially put a microphone and small transducing wires in there and try to get whatever nerves are less to fire, so you get some sense of hearing. So I'm going to take a break. Come back in, say, 20 minutes.